Alright, here we go. This next section is on organic chemistry and biochemistry. So organic chemistry is the study of the carbon atom in all of its glory. Yes, you heard that right. There actually is an entire science discipline dedicated to the study of carbon. And it is just about as unpleasant as it sounds. I took 2 semesters of it in college and was never so happy to be done with the course as I was when I walked out of the final exam on that second semester. This cartoon exactly illustrates how I felt about that class. Hopefully it won't be how you feel about this section. I personally find biochemistry to be a lot more interesting. It's a kind of a weird merger of biological systems and the chemical reactions and compounds that are found within those systems. As a result of this instructor bias, we'll be spending as little time as possible on organic chemistry before moving on to biochemistry. Let's go over some of carbon's special properties. Carbon has 6 electrons total, so 2 electrons in its inner shell and 4 electrons in its valence shell. Because of this, it is able to share 4 more electrons with other atoms to make up to 4 chemical bonds. The bonds are shown by lines between the elements. At the top left-hand side here, a carbon atom is bonded to 4 hydrogens. This compound is non-polar, so it doesn't have a charged end, and so it does not dissolve in water. Carbon can also form double bonds with itself, which is shown by 2 parallel lines between carbon atoms, as in molecule B at the bottom here, or even triple bonds with itself, shown by 3 parallel lines between carbon atoms, as in molecule C at the bottom. Carbon can form long chains where each carbon atom is bonded to hydrogen atoms on the side. These are called hydrocarbon chains, and they are extremely hydrophobic because they are non-polar. Carbon can also form rings with various atoms or molecules attached on the sides. Functional groups are specific arrangements of carbon and other elements that are found over and over again in nature. Each functional group has particular characteristics that will differentiate it from the other functional groups. For this class, you will not have to memorize any of the functional groups, but you should know that they exist and kind of what the term functional group means. Macromolecules are very large, very complex organic molecules that are known to be made and used by living things. Here are a few 3D representations of macromolecules. Catalase, on the left-hand side, is a specific type of protein called an enzyme. Enzymes facilitate chemical reactions, and their names usually end in ACE, so ASE, so catalase. Catalase is an enzyme in your body that helps to convert hydrogen peroxide into water and oxygen. So it's the reason that hydrogen peroxide bubbles when you put it into an open wound. The other example on the right is a small section of a strand of DNA. DNA holds all of your genetic information and is extremely complex. We're actually still trying to understand how DNA works. The 4 general categories of macromolecules in living things are carbohydrates, proteins, lipids, and nucleic acids. On the previous slide, catalase was an example of a protein, and DNA was an example of a nucleic acid. Carbohydrates, proteins, and nucleic acids are all considered polymers because they are made up of the same monomer subunit that's repeated over and over again. Lipids do not have a single monomer unit that repeats, so they're not technically considered polymers. Chapter 2 deals with the biochemistry of macromolecules, and then Chapter 3 goes into some of the practical applications of that with some information on general nutrition. I'm not going to focus too much on the first part of Chapter 3 in this presentation because that information is fairly easy to understand on your own once you grasp the biochemistry behind the macromolecules. The study guide will do a good job of walking you through that part of the chapter, and I put some questions at the end of the study guide so you can kind of check your understanding of the material. Alright, so the first group of biomolecule that we're going to talk about are carbohydrates. So they are also known as sugars, either simple sugars or complex sugars, and they provide the major source of energy for your daily activities. And then they also have a structural role in cells. The more complex carbohydrates can form parts of the cytoskeleton and part of the structure of the cell. So all carbohydrates are composed of carbon, hydrogen and oxygen in a very specific ratio, and that ratio is CH2O. So 1 carbon to 2 hydrogens to 1 oxygen. The monomer subunit is a monosaccharide, and that's a single sugar ring. So remember, carbohydrates are considered polymers because they're made up of the same unit repeated over and over and over and over, and that single unit is the monosaccharide. Here are several examples of carbohydrates. Notice that all of them have a ring of carbons. So in organic chemistry, because carbon's so prevalent, if they don't show an element in a structure, then you're assuming it's carbon. So with these rings, each of the points on the rings are carbon atoms. Monosaccharides have a single ring, like glucose on the upper left hand of the slide. Disaccharides have 2 rings, like sucrose or table sugar, which is on the upper right-hand side of the slide. And polysaccharides have many rings, and the example here is starch on the bottom of the slide. Remember, the monomer unit for carbohydrates is the monosaccharide, so the polysaccharides are considered the polymer unit. Proteins are the next type of biomolecule that we'll talk about. They provide a structural role in cells. They also help regulate chemical reactions in the form of enzymes. They can serve as channels in the cell membrane, allowing substances into and out of the cell. And they can also act as a long-distance signal. They can travel throughout your body to send messages for how the body should respond, what chemicals you should produce, all of that. The monomer subunit for the protein is the amino acid. So here's a generic amino acid. All amino acids are composed of 3 things attached to a central carbon atom that has a hydrogen atom as well. The first thing is an amino group containing nitrogen, so that's on the left-hand side. The second thing is a carboxyl group, and that's one of those functional groups that I showed you earlier, the carboxyl group, and that's on the right-hand side. And then the third thing is some sort of side chain, and that is usually indicated by an R. R kind of stands for the rest of the molecule. But those are the main components of an amino acid. The side groups can be different for different amino acids, and they help determine the amino acid's behavior and characteristics and how they react with other molecules. There are 20 commonly occurring amino acids. Some of those amino acids your body can make, others it can't, and you have to eat them. Otherwise, you can become deficient in those amino acids. The amino acids that you have to eat that you can't make are typically called the essential amino acids, and animal proteins tend to be complete, meaning that they contain all of those essential amino acids, whereas plant-based proteins are not usually complete. That's one of the reasons vegetarians have to be very careful to eat a diverse group of vegetables to get all the protein they need, because there's no one vegetable that's complete like you would see with an animal protein. Many amino acids join together through peptide bonds to form a polymer called a polypeptide. So the polypeptide can then fold into various arrangements, such as the beta sheet that's shown here, by hydrogen bonds. So both peptide bonds and hydrogen bonds are very important in the structure of amino acids. Some proteins, such as enzymes, have a 3D structure that must be completely correct for the protein to work correctly. This picture is of hemoglobin, the protein in your blood that binds oxygen to carry it to your tissues. Each of these spirals is made up of many, many polypeptide chains that are folded together. Lipids are the next group of biomolecules that we'll talk about. There are 3 general types of lipids that are important. Fats, which are used for long-term energy storage, steroids, which are chemical messengers in your body, and phospholipids, which are the primary component of cell membranes. Fats are made up of hydrocarbon tails that are attached to a glycerol molecule. So the picture here shows 2 fatty acids. The one on top is considered saturated because it has no double bonds between the carbons. The fatty acid on the bottom is unsaturated because it has at least one double bond between the carbons. Notice that the double bond gives this tail a slight kink, a slight turn. That will actually be important later. The red part on the left-hand end of each molecule is the glycerol component. The black carbon chain with hydrogen atoms attached is called the hydrocarbon tail. In general, most nutritionists believe it's better to eat unsaturated fats than saturated fats. Unsaturated fats are found in plant sources, like olive oil, and saturated fats are found in animal sources, like butter. Because of the kink in the unsaturated fatty acid, unsaturated fats are usually liquid at room temperature, whereas saturated fats can solidify enough to be solid at room temperature, like butter. Steroids are composed of 4 fused carbon rings that have various side chains attached. They are very important messengers in the body, and they can actually have drastic effects if they're produced in excess or not produced enough. So, for example, there's a disease called hyperadrenocorticism, where your body makes too much cortisol, and it can cause you to lose muscle mass, you drink a lot of water, you urinate excessively, you become pot-bellied, and you can lose hair. So the picture at the bottom right-hand side is actually of a dog that has hyperadrenocorticism. Phospholipids are the last lipid molecule we're going to talk about. Here are 3 different representations of a phospholipid. They look very similar to a fatty acid, except that they have a phosphate head group that makes them have a polar end that is hydrophilic. That's really important. So the tails are nonpolar and hydrophobic, but the head is polar and hydrophilic. One of the tails is unsaturated and has a kink in it. So when we talk about cell membranes later, we'll mention this again because phospholipids are the primary component of cell membranes. The last type of biomolecule that we're talking about today would be nucleic acids. There are 2 types of nucleic acids, DNA and RNA. DNA stores genetic information, and RNA is used for cells to synthesize proteins. The monomer unit of a nucleic acid is the nucleotide. So a basic nucleotide structure is shown here. A nucleotide is made up of a 5-carbon sugar, a phosphate group, and a nitrogen-containing base. DNA has 4 different nucleotides. The names of those nucleotides are adenine, guanine, thymine, and cytosine. The one here on the slide shows adenosine, so adenine, and these are all indicated by kind of the first letter of their name. So adenine is usually indicated by an A, guanine by a G, thymine by a T, and cytosine by a C. The general structure of DNA is a double helix. The bases pair up in the middle of the helix through hydrogen bonds. Adenine always pairs with thymine, so A goes with T, and cytosine always pairs with guanine, C goes with G. The backbone of the structure is composed of alternating phosphate and sugar groups that are joined through covalent bonds. Notice that adenine and guanine have 2 rings. These are called the purine bases. Thymine and cytosine have 1 ring each, and they're called the pyrimidine bases. Because of the base pairing rules, so A goes with T and ends with G, the width of the DNA helix is uniform. There aren't any bulges. The strands of the helix arrange so that the nucleotides face up on one side of the helix and down on the other side of the helix. For this reason, the strands are said to be anti-parallel. We'll actually discuss DNA and RNA in more detail later in the semester when we go over genes and protein synthesis in the cell. Alright, so that's the end of the second section. The next section we'll talk about is going to go into cell structure and membrane transport.